Okay. Um, I'll start then. Hello everyone. Welcome to the fifth, I think, meeting for the USC Journal Club. I don't know if the professors can make it today, but they probably can. Hopefully they'll join soon. If not, we can just lead a discussion by ourselves. But this week's paper was on something I think is really interesting, and I know this week's paper had a lot of I mean, every paper does, but it had a lot of jargon. And this week, three members stepped up to be part of the slideshow. So I kind of gave kind of bigger roles to them. So during the slideshow presentation, hopefully you can hear a lot from them, learn a lot from them, and yeah. But I'll start first off because I'm doing the introduction for this slides. So I'll start screen sharing in 1 second. Sorry, 1 second. All right. Can everyone see my screen? All right. Awesome. So, I'll kind of just go ahead and start. And then the members who contributed, if when I get to your section, just like last time, you can start presenting. Say next slide if you want me to go to the next slide. And yeah, you can take some more time today, because you have bigger sections overall. But this week's research paper was on in vivo cyclic. Oh, I'm not going to read the whole title. It was on Yamanaka factors and how cyclically induced Yamanaka factors can have really, I guess, impressive potential therapeutic possibilities on on the brain. So here's a table of contents, and I'm just gonna go over the introduction and kind of go over the basics of the paper. So yeah. Um, So these are the main four things I wanted to highlight about this paper before we dive into it. One is obviously Yamanaka factors. Now, Yamanaka factors is not one thing, it's a set of four different transcription factors that have been that have all been shown to kind of be able to reverse a differentiated cell back to basically a stem cell or an iPSC. So induced pluripotent stem cell, I think. And this is, if you know anything about stem cells, you know it's like a very controversial but widely known possible therapy for like a bunch of a whole vast amount of diseases. So just this idea that there's something that can change a differentiated or fully mature cell back into a stem cell is crazy. And I forgot the exact name. It was like something socks. Anyways, the four different transcription factors, um they have like an acronym for OKSM, I think. And the paper kind of referred to them in that manner too. So yeah. And then yeah, see obviously there's huge potential therapeutic possibilities for the Yamanaka factors um on neurodegenerative disease because once you have a stem cell, you can say you could differentiate it back into a new neural cell, you're completely getting a new neural cell. And that's basically the best thing. I mean, not the best thing, but basically one of the best things you could do to combat neurodegenerative disease. Um, Tumor formation has been kind of the others kind of bad end of Yamanaka factors and any iPSC formation in general because well, first of all, stem cells themselves they grow really fast and they're not as regulated. So they have the implication of forming tumors. And also Yamanaka factors in general, they kind of have they have been known to have things that include oncogenes, so basically genes related to cancer, and that's not good. So many cells have been seen that with too much Yamanaka factor expression within the cell, they often turn into like, or like when you induce too much Yamanaka factors into mice, they often grow giant tumors in their brains, and that's not good at all. Um, but then where this paper kind of comes in is with that cyclic appeal. So instead of constantly inducing Yamanaka factors 24/7, because that would almost result in, like cancer. The cyclic idea they have is like kind of like cyclically inducing Yamanaka factors, not constantly, but every now and then. So it could be like a balanced bit where the cells are able to kind of rejuvenate, but not, form tumors or whatever. So yeah, that's the overall background. Hopefully that can help you kind of get a baseline before we dive into more. But yeah, here are the methods. Samian, if you're ready to present, you can start talking.
All right, yeah, you can move on to the next slide. Awesome. So in this study, the researchers used 49 male and female mice. They crossed mice with I don't really know how to pronounce it other than just say the letters. It's the alpha CAMKII controlled transcriptional transactivator with the mice with the doxycycline inducible OSKM code. And this resulted in a double transgenic mouse model with neuron-specific Yamanaka factor expression. And this means that in the absence of doxycycline, the mice expressed Yamanaka factors. And so they started treatment when the mice were 6 months old, and then until they're 11 months old. So the treatment was for 5 months. And they had two groups. One was the cyclic group. So in the cyclic group, they had doxycycline removed in the mice for 4 days per week, and then restored them in the other three. But with the continuous group, which is the second group, they removed it for 4 months continuously. And so those were their two test groups. And they had a control group, which were mice without the transcriptional transactivator gene. And doxycycline was just used to inhibit the Yamanaka factor expression. So that's why it was removed. Our next slide. So the second part was their treatment analysis. So they gene expression was actually determined by qPCR. There is like a little bit of a enlarged form of qPCR. I don't really know how to exactly say what each letter means, but so they also performed RNA sequencing on the hippocampus and neocortex tissue. And this identified differentially expressed genes, which were linked to memory, plasticity, synaptic function, and aging. So basically, they used RNA sequencing to see which genes have a different expression. And then they used behavioral tests to further analyze their tests. So they had three tests that they mentioned, which were the open field test, novel object recognition test, and the Y maze test. These tests were used for either memory or anxiety and depression levels. So the open field test, they had mice analyzed in a box. If they were not sticking to the sides and instead going towards the middle, that means they're actually showing an age-reduced form of anxiety and depression, which is less. In the novel object recognition test, they put mice in a box with object A and object B, and then they took them out. 2 hours later, they put them back in, but instead of object B, it's a different object. So only object A is the same. And so this test actually allows them to measure memory if the mice went towards object A. And the final test they used, which was a behavioral test, was the Y maze test, which they put mice in mazes basically, and then they allowed them to roam around and put them back later, and that was how they measured spatial memory of the mice. So these are just the ways they analyzed their testing, and it showed that cyclic grouping actually helped with functioning and reactions. That's it.
All right, so I'm doing the results. Next slide, please. So there are kind of a lot of like key findings to this research, and one of them being the neuron-specific programming part. So the YFs were activated only in certain brain regions, like the hippocampus and neocortex region, due to the AKM and K2 promoter that blocks expression to neurons in charge of the memory and learning. And there was also no YF activity detected in the cerebellum. And the next one being the gene expression changes, the RNA sequence showed that in the research 1,419 DEGs in the hippocampus of the younger mice, and 604 genes decreased while 815 genes increased. And these changes were linked to aging-related processes and epigenetic rejuvenation through increased H4K2 20Me3 methylation in neocortex and hippocampus CA1/CA3 regions. And there was also safety findings like high mortality in mice with YF from birth. Long-term YF exposure may be harmful. There was also no deaths in mice treated starting at 6 months, indicating age-dependent, so like adult mice, and no tumors that were formed, so no stem cell state. And the cyclic exposure, researchers found that cyclic exposure, which they put 4 days on and 3 days off, worked as planned by using the immunofluorescence. It was also safer for the adult mice, as stated. Next slide, please. There were also behavioral improvements shown in the mice, like the cyclic YF-treated mice showed more youthful behaviors, for example, less anxiety in open field tests, better novel object recognition, and improved Y-maze performance. Continuous treatment had no effect. And then the neuron-specific gene changes, the RNA sequence identified 94 DEGs in the adult mice, 70 decrease and 24 increase in dendrites, confirming neuron-specific reprogramming. The neocortex showed stronger changes than hippocampus, and the zinc finger proteins were one of the key regulators. And enhanced memory circuit activity, the treated mice had more CFOS and hippocampal memory regions post-testing, and lots of the KLF4 reduced CFOS, explaining why cyclic dosing was better than continuous. And lastly, the ECM rejuvenation. The treated mice actually exhibited reduced aggregate and fewer PNNs and neocortex. That's not the hippocampus, like youthful brain flexibility. And the Western blot lab test just confirmed these findings. And that was it.
So I'll be going over the discussion. Next slide, please. So this paper is the first study to have inducible Yamanaka factors or YF expression that led to successful neural restoration. This is also the first study to validate that neurons don't completely revert to stem cell state as a result of YF expression, and this is still kind of in the progress of being researched as to why. This study also confirmed that YF didn't form any harmful tumors in mice. Mice given cyclic YF or repeated cycles experienced cognitive enhancement, while mice given continuous YF didn't experience any change in behavior or cognition. Researchers observed gene activity related to neural repair, supporting the effects of YF expression, and memory cells were repaired during partial programming. Next slide, please. Continuing on, there was a decrease of a protein called aggrecan, which is a protein that's found in the extracellular matrix that plays a role in structure and support, and this is correlated with memory enhancement. There was no significant change in DCX levels, and this led to the conclusion that YFs do not significantly impact neural growth and differentiation. DCX is basically just immature neurons that express the DCX protein, which play a role in neural migration and differentiation. Partial reprogramming of neurons led to a generalized increase in histone methylation. And then there's also a strong relation or a strong correlation between cyclic expression of YF and age-dependent cellular and molecular changes. And lastly, all of the changes seen from cyclic YF expression could have potentially contributed to memory enhancement. The reason that the extracellular matrix was like kind of mentioned in this paper was that it has a role in synaptic elasticity and neuron youthfulness. It supports neural structure and influences cell behavior and regulates neural activity by regulating ion channels and expression of neurotransmitters. There's also an ongoing theory that manipulating the levels of some epigenetic markers can extend the lifespan by increasing genome stability. And Epigenetic rejuvenation of neurons through partial programming can reverse the decline of aging-associated markers like H4K20Me3 through remethylation. Next slide.
All right. 1 second, let me stop sharing. Okay. So, because we have a lot of time left, I kind of want to get more into discussion today. Um, and specifically depending on the specific section, I have some questions. I know like Sami and you prepared questions too, Kailey. Mahita, I don't know about you, but if you prepared any questions, ask. And everyone else, like the members, you can try responding through the chat. That might be easier because these are like questions to everyone. So we can kind of get a gauge on what everyone thought. But yeah, like Samian, I know you had a question. Do you want to you can ask everyone and then y'all can answer through the chat.
Yes, thank you. My question was, if it's related to how they did the methods, kind of. If you could rejuvenate one part of the brain in humans instead of in mice, what would you choose? Like you can use like prefrontal cortex, hippocampus, etc. What would you do? What would you rejuvenate? And that's my question to you guys. You guys can send your answers in the chat once you have them. If I could like add on to that, I think I see that like obviously the common answer here is hippocampus. So I actually want to kind of direct a question to Professor Rust. So obviously we know that hippocampus is like a big part in neurodegenerative diseases like Alzheimer's. So if we were to rejuvenate the hippocampus, is it possible to like um regain a lot of memory? Like how like would you regain memories from like let's say childhood and stuff like that that you wouldn't even have normally as like like a person without a neurodegenerative disease? Like let's say a normal person would not remember things before the age of 3, and then a person the neurotypical or sorry, a neurodegenerative disease person um, happens to have their hippocampus rejuvenated, would that then increase their memories beyond a person who is, not rejuvenated, or would it just improve it slightly, like underneath a person who does not have a neurodegenerative disease? Sorry if that was a little confusing.
I think Professor Russ might not be able to talk like on mic, but he can answer through chat too. Sorry about that. Professor Russ, you can just answer through the chat if that's easier for you too. And also while he's doing that, I kind of wanted to, I guess this is another question I wanted to ask. Um, I'll just ask it right now. But I found like a lot of the methodologies, especially like for looking at specifically like behavioral, looking at it from a behavioral standpoint, I feel like in a lot of neuroscience, there's either like a top-down approach or a bottom-up approach, where a bottom approach would be more like you're looking at the specific biomarkers. But in like this case, in the methodology, there was also like more top-down approaches as well. Because like for the behavior and like anxiety levels. They like put rats in like the box and then did like the box test and then like kind of did like qualitative analysis kind of, but they also use like I guess statistics a little bit to see like how much anxiety the mouse showed from that, just like from looking at them running around in a box. And I thought that was really interesting. So if y'all have if I guess my question would be, what was your what did you think was the most interesting methodology that was mentioned in this study? Also, Samian, you can just ask your question in the chat. I think that might be easier for Professor S. Or like directly send it to him. All right, sorry about that. It's like really messy, but I try to copy and paste it from Discord. I can read aloud some of the main questions, and also if you have like any feedback or if you have any ideas on like anything related, any answer or anything like discussion based off these questions, you can feel free to answer. But I guess maybe this is kind of branching off what Samian just said. I think this might have actually been your question, but like if it's basically this question. If turning on certain genes in older brain cells can make them act younger and improve memory, what would happen if we actually were to increase the number of new brain cells being made in the hippocampus? What would be the effects? This is kind of similar to your question, I feel like, Samyon. So, I guess this entire concept, if anyone has any thoughts, you can try answering. I think Professor Russ is going to type something up too. So,
I want to try to answer the question. Let me see whose question that was really quick. Um, I believe it was before mine. I think Lovea. I think that's
No, it
sorry if
was
that's
my question. Sorry, I didn't change my name in the Discord.
oh sorry, Bavavia, sorry. Um, so that question, um, it doesn't really say how to do it. Like my question was like, is there a way to do that? But your question is more like theoretical, and I like that. So I think what would happen, since this is neurogenesis, it's basically what happens with the mice. I think it it just does that on a higher level. I feel like that would just enhance their memory, I guess. And I feel like especially in younger people, it would reduce like stress responses when we try to remember, I guess. And then I think that's just it. I think it helps more with memory. So I'm not sure if that is possible, but if it is, I feel like that's exactly what would happen. It's like how it would happen in mice, where it we show age reduction in our functioning, like processes like remembering things. I feel like that would happen on a higher level. Again, I can't, that's just my like it's kind of like a theory, I guess, but that would be my answer to that.
Okay.
Just to give an answer.
Yeah.
Flavia, do you have any like after like going over everything, like as like through the slideshows, do you have any like updates or like potential ideas on the answer to your own question?
I agree that it would probably like improve memory, and I was basically thinking the same thing that Samian said. So,
Um, I think that like if we made like to answer this question, if we made more new brain cells in the hippocampus on top of making like the older ones act younger, it'd probably like help the memory even more, kind of like what she said. But like new neurons are supposed to be really good at forming new connections and learning things. So having both the young acting old cells and like the brand new ones, it sounds like it could be really powerful. But I also feel like if you had too many neurons at once, it could mess things up badly and like the brain wouldn't know how to fit them all in or wouldn't connect right. So I feel like it could help a lot, but like it needs to have a balance, if that makes sense.
Yeah. And something I also wanted to point out was essentially like the hippocampus is really tied with memory, but the brain isn't like, the main I feel like one of the most crucial things you got to know about oh one of the most misunderstood things about neuroscience is that like the hippocampus is for memory, the amygdala is for emotions. It's not separated like that. It's more like a spaghetti bowl. So when we say like specifically regenerating neurons in the hippocampus, we got to think about like how these neurons protrude to the amygdala. And when you think about it, like when you have memories or when you like remember something, even like when you hear music or smell something, it's always tied in with memories. And these memories also have really deep connections with emotions. Um, and so many different facets. It's not just like one thing. We're not robots with, like different like places, separated for these specific functions. It's all really interconnected. So the concept of this question is something that you can explore really well, but when you think about it like from a perspective like is regenerating neurons in this hippocampus specifically going to change memory? We wouldn't know because it's just so nuanced.
I actually wanted to add on to that, kind of redirect another question to Professor Ross. I feel like I'm overloading him. I'll type it in the chat after, in case he doesn't hear it, because I'm pretty sure he's answering some right now. But with what you and Dithya said, with if we do further rejuvenation in the hippocampus and some old cells are like, or sorry, old neurons are either rejuvenated or something goes wrong or like they're removed completely and we just neurogenesis occurs, we add new neurons, get rid of the old ones. How would that affect memory? Like obviously the memory would be better with the newer neurons, but those past neurons which had all these tight and deep connections, these like very like long connections, and those connections are what we rely on to remember things. Those connections are what memory is, basically, those neuron connections. So if we get rid of those neural connections, the older ones specifically, and add new ones, what does that mean for our memory? Do we forget certain things? Do we remember things better? Do we have this memory of like we remember this specific event better, but we remember this specific event less. So I'll ask that in chat to Professor Russ as well, but that's just like something if anyone else knows anything about that, just to add on to the discussion. So
Does anyone here know anything about engrams? Okay. If not, I can give like a basic explanation. I think it kind of ties in with Samian's question. Like Samian, if you know what engrams are, you can try explaining it too. All right. So they're kind of like um it's like it's basically the concept that your memories are stored in more like a pattern of neurons rather than a single neuron. So a lot of people found that in mice, when they're going through a maze, specific, not just a specific neuron, but specific patterns of different neurons light up in a certain order and stuff like that. And these engrams encode for their memory. So what you kind of said about if a neuron is degenerated or blah blah blah, and how that might affect a single memory, if it's part of like an engram, it could have a much wider effect. You know what I mean? But I'm not an expert in that field. It does sound really interesting though. But yeah. Also, I think Mahita also had a question. So, if you're comfortable with sharing that question, Mahita, you can also ask your question as well.
Yeah, so mine was like a very generalized question, and I'm pretty sure like one of you can answer it. But like it's pretty well known that like even though there's like a very textbook diagram of the brain, it's like the anatomy of it is very different from person to person. So I was asking because the anatomy of the brain is so diverse, would rejuvenation of neurons occur in the same way in like both hemispheres of the brain?
If you have an answer, also feel free to answer in chat. All right, Jaden, you can I see that you have your hand raised. You can try answering.
Experiments using them, like what sorts of techniques they use.
And so I couldn't hear you. Could you repeat that?
Yeah, so I was just wondering how engrams are like studied in like different experiments and like how they're like, I guess, kept track of or like, yeah, like used in experiments, I guess.
That's a great question. I can try answering, and then anyone else who has any knowledge on, I mean, I guess not, but I think it's interesting, so I'll try answering. I know that one research study, they kind of looked into the neurons of mice, and when neurons fire action potentials and like transmit neurotransmitters onto the next neuron, they open I think yeah, calcium. Wait, calcium or chloride? I think calcium receptors on the neuron, right? So basically what they did was they tagged these calcium receptors and it kind of light up every time a neuron fired an action potential. And then they placed the mice inside of a maze. And as the mice were going through the maze, the researchers could kind of track what neurons were firing visually because the neurons would light up when the calcium channels opened. And that's actually a really, really cool, like ingenious way of how they could track it. And this is also really amazing because it would be shown through visual representations, right? Like this neurons would light up and then you could kind of like map that out on the brain. And that's how you visualize engrams. They're like these specific patterns through in the brain. And like every time the mice were in a specific section of the maze, the same engram would light up. And that's just something really I thought it was really interesting. But yeah. Um and I also kind of wanted to address Ender's question. Um I think Samian kind of answered it. But wait, Samian, do you want to kind of talk about how you answered it first?
Sure. Let me really quickly read the question a little better. Okay, so yeah, with my question again, it's more like I'm not really an expert in this field, so it's more of my, I guess from what I read, what I theorize. I think that it definitely would enhance their PTSDs. I guess at least the memory part of the PTSD. Obviously, I don't know about their like the effect of it, like how would they would act in turn due to the PTSD. But they would definitely have more of a reaction to that. Like they would have that in their memory because obviously the what's it called? The memory is enhanced now with the neurogenesis or the rejuvenation in the hippocampus. So it would definitely cause their memory to be better and enhance them to think about the PTSD. I'm not sure how their symptoms would be affected, but that would definitely happen. And I see that your hand is raised, Ender. I don't know if you want to respond to me or ask another question.
I just wanted to clarify like the main thing I was asking because I realized I didn't word it very well. So I studied psychiatry as well for a while, and I'm still studying it along with neurology because I'm trying to want to do. But with that, it's with a lot like more, I was gonna say war based because that's kind of the main thing I was thinking of, like big in the moment, not complex, but in the moment PTSD. The flashbacks are when they remember exactly where they were. But when people get older, a lot of the time, those parts can get a little more blurry. So I was wondering, do you think it would trigger the really bad flashbacks and make you feel like they were actually there as more than it was before as they were slowly forgetting it?
I'm not exactly sure, because I didn't really look at the results part. I mean, as much as I did with methodologies, but I think that it definitely would cause them to, if they do have like something like triggering that, because like as Sun was said, it's not just like them remembering. It's like like an experience, like let's say the music or like the ground, the location they were at, or like the certain smell. I feel like that would definitely cause them to have enhanced PTSD like effects, like repercussions. Like like you said, like an intense flashback. I feel like with the rejuvenated hippocampus, I feel like that definitely will happen because these neurons are obviously they're age reduced, right? They're going back kind of to their state like they were, let's say 20, 30 years ago. I mean, obviously that's not exactly the case, right? That's more like it's just like an analogy, I guess. It's not exactly going back to how it was 20 years ago, but it's definitely, it's renewing itself. It's not like as worn as it like I guess in a sense worn as it was before. So I do think that yes, it would cause more intense flashbacks from what I read. I think that that would happen. And that's a good question, actually. Um, Again, I'm not completely sure, but that's what I believe. So,
Awesome. I honestly have no knowledge in that specific field, but I do know that PTSD, if y'all know anything about epigenetics, I mean, that was one of the results of this study, right? So epigenetics kind of showcase that how your environment might impact your genes. And through that, PTSD could genetically alter you. So that there's definitely implications regarding memory, like all even like Alzheimer's relating to PTSD. Kailey had like a question for all of you, kind of like Sami and Mahita. Y'all can try answering in the chat. Kailey, do you want to ask your question out loud?
Oh yeah. So my question was like about the results from this research paper I talked about. Which one do you think is like the most impactful one and why?
I'll give everyone 2 more minutes to answer this question because I think it's just a really good way to reflect on everything. And I think what everyone's hitting on is basically kind of all the fact things that I found really interesting about this paper. Um, and yeah, the concept of rejuvenation is really, really interesting. The more you think about it, the more kind of like I guess ethical it gets. Even like what Ender said, um, it kind of gets to like a point where like you think about memories, yeah, and you can like think a lot about that. I also kind of want to address Jolse's question. Can rejuvenation theoretically be used to erase trauma because of restoring the neurons forgetting the main event that caused a traumatic experience? Um, this is kind of like exactly the example of what I was talking about when I said like keep if you think about like this idea a lot, it kind of gets like into a more philosophical stance or like a perspective. But yeah, it's kind of interesting to think about. Um, I don't have an answer to that question, but if y'all do, you can probably text it in the chat. Um, what I wanted to go over for like the last couple like 10 minutes is I wanted to go kind of get into like I guess the first parts of the capstone project that we're going to be doing because y'all have been asking me a lot about that and I feel like I didn't address it. I mean, I have been addressing it a lot because we're doing these meetings, but next meeting is going to be like the last paper meeting and then after that we're going to start a capstone project. So obviously I'll talk about it a little bit more next meeting, but I kind of have y'all can feel free to ask questions about the capstone project in the chat and I'll try to address them right now. But what I was planning on doing like last year was after meeting six, um you're gonna have like basically we've gone through read through a lot of articles. So you're gonna get your chance to choose a specific topic from neuroscience and basically you can either do like last year, Tej restricted it to like a spotlight article or like a literature review. But this year, I wouldn't mind like you maybe coming up with your own research idea, writing an abstract, and sending it to me. And you like obviously if you have enough resources, you can do a whole research. But like the capstone project is meant for you to figure out your specific niche in neuroscience, and kind of pursue your own research and start looking at the gaps in literature your own self. So that's basically what I'm planning on for the capstone project. So either you can do like a spotlight article, that's basically where you read one article and like do basically like a review on that article. A literature review, which is way more work. It's like you read through like a whole slew of research in one category and then kind of produce a paper that is talking about the state of that subtopic in neuroscience. And it kind of like covers all the different um I guess all the different facets of that specific facet of neuroscience. And then for research, you can either submit like an abstract to me Or like if you want, you can do like a whole research, but that's going to be really hard, especially since I'm only going to give y'all like a month. But um like if you do choose to submit an abstract, you can always, talk about the abstract for this club, and then you can continue on with that research after this club ends, and do like symposiums or whatever. But yeah, for now, um start trying to think about like all the papers we've read and any other neuroscience knowledge you might have, um, and try to see what interested you the most, what was the most fascinating to you, and what you think you could genuinely pursue for like maybe a month to kind of get into, lock in on, and start reading. Because as you know, you've probably figured out from this club that reading scientific literature is not easy and it's not fun all the time. But if you do find like if you have the baseline of neuroscience, then you can find like a specific point where you do really find genuine interest in that specific part of neuroscience. And the more you get into it, the more rewarding it will be. So that's basically what I'm trying to encourage through the capstone project. I'll talk about more like details next class, but honestly, I'm not trying to restrict y'all into doing anything specific. It's creating something of your own work based on something you found interesting. So yeah. Does anyone have any questions on the capstone project? Jaden, I see you kind of have your hands raised.
Yeah, so is it a literature review or is it not? Like it sounds like we are like just looking through a bunch of articles and like compiling them into one paper, or are we like making our own hypothesis, I guess, and then kind of figuring it out?
I mean, you can either do a literature. I mean, if you want, you can do a literature review, or if you want, you can start your own research. Like, I'm not restricting you to a specific type of thing. It can be anything from just reading one article and writing a review off that article, all the way to conducting a full-blown study with like university professors or whatever. But I just want y'all to all find that one, gap in literature where you're interested in. And if you were to start a research, then you typically have to do a literature review first. So yeah, that kind of does mix. But yeah, if you want to do research, feel free to, and then you can like present your abstract or present your poster or whatever during the symposium. Yeah, Samian, if you have a question.
Yeah, I asked in the chat, and someone else seems to like agree, like want to ask the same
Oh,
question. I'll just
okay.
ask it. I'll ask it again here. Like, could our research tie into general neuroscience, but also like other topics and subfields like psychology or like psychiatry? And like for example, like research in like disorders and like disorder behavior and like studies with that. And like, does it have to tie into what we read, or can it be like external as well? Because from what we read more like neurodegenerative diseases. It's like really sub like into that niche. So we go beyond that, like into disorders, psychology, stuff like that.
Yeah, honestly, I'm honestly more into like psychological disorders as well. So, yeah, I'd love it if I saw some of those projects. You're not restricted at all by the papers we've read. I just kind of started off. I kind of try to incorporate these papers first off because the professors are really into these. Like these are their specialties, and also these papers are like I think the best view of an intro into neuroscience. But yeah, if you have like ideas on like more behavioral disorders, psychological stuff, feel free to conduct research, do literature review, whatever you want on those topics because I honestly love that part of neuroscience as well. Mahita, if you have a question, you can.
Yeah. So when we're like towards the end of it, like once we've like finished our capstone project, are we gonna like submit a recording to you and then you choose like which ones we submit like in a meeting, or like how do you like predict that will work?
Okay. So I think it was in my calendar. I forgot when. It's gonna be like so we're gonna start after next meeting, and then around like early September, I'm gonna have a check-in meeting with y'all, and then towards like the latter half of September, well, like mid-September, you're gonna start submitting your abstracts or whatever, like your presentations and your papers. And then after that, I'll schedule like a one-on-one meeting with you where I time you and I record your presentation basically. And that's gonna take up basically the entire latter half of September. I'll just be going through meetings with all of y'all, but I'll be recording you in on Zoom. I feel like that's the best way to do it. Um, I mean, you can try recording yourself, but doing it in like just one go, that's what I honestly recommend to everyone instead of just like sitting down trying over and over.
And
And
do you
so
think we're All right. Do you think there'll be a time limit? Like, what do you have an idea of what the time limit might be?
Last year I had to present my literature review in 10 minutes. So yeah, a video would probably like I'd recommend you to do it under honestly, I'd even make it shorter because if you have the presentation and like the paper ready, then if I were to make a symposium, I wouldn't want like the video to be really long. I just want you to do like a basically like what they call an elevator pitch in the video where you do a quick run through of your research or your literature review or whatever. And then the people who are interested can actually look into your presentation, look into your paper on the website. So yeah, don't expect it to be too long. Like honestly, under 5 minutes. That's what I'm trying. That's what I'm thinking about right now. Samian. Mhm.
For our capstone project, well, let's say we do a lit review or we start our own research, whatever. Like let's say in our next meeting, would you like kind of guide us through how to get started, like tips and tricks on how to like do these presentations, how to actually, start with literature reviews and like, actually start with starting our own research. Like, would you like guide us through or like do we have to find those resources on our own or that's just my kind of question.
Uh, yeah, I mean, I can definitely give you heads pointers. Um, as like I think next meeting we'll go over the paper and then I'll give you more pointers on like specifics on research and literature. But if you have any specific questions, just email me. I feel like that's the most efficient way to directly address your whatever you're doing. Um, yeah, so feel free to email me. I'm trying to work on research right now too, and it's hard. So, yeah, I'd love to help. Um, I think if that's all the questions we have, then we can end the meeting for today. Sorry, it was a little chaotic with all the discussion. And the professors, I don't think Professor Winston could make it. Professor Rust was on his phone. So, um, if you had any questions that weren't answered, just you could honestly email them to Professor Rust. I know he's down to like answer questions through email. So yeah, you can do that. Thank you everyone for showing up. Next meeting is our like last research meeting, and I'll also be going over um going deeper into like the logistics for our capstone project. So yeah, see you next week. It'll be in 2 weeks, same time. So yeah, thank you. Also attendance form. 1 second. Sorry about that.